So, okay, dear students, I told you in the last video that there are two types of strains in Streptococcus pneumoniae. It was observed by Griffith. One is smooth strain and another one is rough strain. He observed two strains. One is smooth and another one is rough strain. And smooth strain was virulent and the rough strain was non-virulent. What he did in his experiment was like this. In his experiment, he took a S strain bacteria, S strain Streptococcus pneumoniae, and he injected it into a mice. Injected to a mice, and obviously it was virulent. Mice died. Then R strain, he took R strain and injected to a mice, and mice was alive. Mice alive. Then he took S strain, but what he did, he heat killed it. He boiled it and he killed it, and he injected it to the mice, and mice was alive. Then he took the mixture of heat killed S strain and live R strain, mixed two bacteria, and injected it to the mice, what he expected, mice should live happily because he killed the poisonous content. He thought he killed the poisonous content of a strain, and live strain is not harmful. So when he combined these two and he injected to the mice, the result was shocking, that the mice died. He started thinking about the reason why the mice die, even though he used heat killed a strain. Then he thought that, conclusion, there might be a transforming principle. There might be a transforming principle transferred heat killed S strain to R strain, and it enable R strain to synthesize smooth polysaccharide coat and become virulent. And the transforming principle transferred the virulent quality of S strain to R strain. But he was unable to tell what was actually the transforming principle. what is the material exactly? He was not able to explain it. So he explained that there might be a transforming principle. There might be a transforming principle transferred the virulent qualities of a strain into non-virulent or strain. It transferred some content of a strain is able to transfer the non-virulent R strain into a strain. But he was not able to tell what exactly it is, and he also concluded that the transforming material must be the genetic material. Must be the genetic material. So I'll repeat his experiment once again for your convenience. He took mice and Streptococcus pneumoniae bacteria. In Streptococcus pneumoniae, he observed two varieties. One is virulent variety, and he named it as S strain with a smooth coat, and non-virulent variety, he named it as R strain with the rough coat, rough outer membrane. Then what he did, he took S strain and he injected it to the mice, and he observed that the mice died. Obviously, because S strain is virulent. Virulent means harmful. When he injected that harmful substance to the mice, automatically it dies. Then what happened? He took R strain and he cultured R strain bacteria and he injected it to the mice. The mice was happy and it was not shown any abnormalities. Then he slightly changed the experimental methodology. He took the heat killed, that means boiled S strain, and he mixed it with the R strain bacteria, and that combination he injected, and what he expected, anyhow he boiled that S strain, so it died. There should not be any impact of it on the mice. But the shocking result was the mice died by taking that injection. Then he started thinking what might be the reason. Then he concluded that the transforming principle, something is there that has been able to convert the non-virulent, remember my words, the non-virulent R strain into virulent S strain. Some material, some unknown thing is able to transfer the non-virulent R strain into virulent S strain. And he also concluded that that particular unknown transforming principle will be the genetic material. That unknown transforming material will be the genetic material. This was the conclusion. And if they ask, explain Griffith's experiment, you should write the following things. First, you have to write, he took mice and Streptococcus pneumoniae for his experiment. And in Streptococcus pneumoniae, two varieties he observed. Number one, smooth virulent variety. This is having smooth mucus coat on its surface and it is poisonous and it causes pneumonia to the organism, infected organism. And the second one is R strain. It is with rough surface but it is non-virulent, not harmful, and it does not cause us pneumonia when it is injected to the organism. Then you should explain the experiment. In first setup, what he did, he took arstrine and injected it into the mice. Obviously, mice was attacked by pneumonia and it dies. In the second set of experiment, he took the arstrine, obviously it is non-virulent, and injected it to the mice, and he observed the mice lived happily without any infection of pneumonia. Then in the next step, he used boiled or heat killed estrine and mixed it with arstrine and injected it to the mice. And what he expected, mice should live without any problem, but he observed the pneumonia symptoms in the mice and it dies. This result was shocked because it was not expected by him, and he started thinking what might be the reason for this, and he was able to conclude that something is there, some transforming principle, he called it as, is there that has been able to convert the non-virulent arstrine into virulent estrine. And he also concluded that that particular transforming principle is the genetic material. So if you explain this much, it will be more than sufficient for 5 marks. Next, biochemical characterization of transforming principle. Then people started asking about what is that transforming principle? What is the chemical nature of it? So he was unable to give the answer. So some other scientists, they started analyzing it. Biochemical analysis of transforming principle. Biochemical analysis of transforming principle. Now, it was a job of scientists to analyze what was it exactly. So, following scientists, Oswald Avery, MacLeod McCarty, McCarty, and then what they did, they worked on transforming principle. Transforming principle. What they did for it, they purified biochemicals, that is protein, DNA, RNA, from heat-killed S cells using suitable enzymes. What they did, they purified DNA, RNA, proteins, carbohydrates, from heat-killed S cells. Heat-killed S cells. Then what happened? Using suitable enzymes. Using suitable enzymes. Now, you should confirm one thing, that purified means what? Purified means separated. they separated DNA from the heat-killed estrine, they separated RNA from the heat-killed estrine, they separated proteins from the heat-killed estrine group bacteria, and they separated carbohydrates from the heat-killed estrine bacteria. Then what they did, they cultured or they took a heat-killed estrine and proteases which are able to digest the proteins, and ostrine, live ostrine, and injected to the mice eyes. What it indicates? It indicates that proteases, even though they were able to digest the proteins in the heat-killed estrine, the estrine transforming principle has transformed live ostrine in non-virulent, live ostrine into virulent form. That's why the mice, when it was injected, it died. That means proteins are not the genetic materials. Then heat-killed estrine, RNAs, which digest RNA, live ostrine, injected to the mice dies. This experiment confirms that even though RNA was digested in the estrine, boiled estrine, heat-killed estrine, the transforming principle was not died. It was able to convert the non-virulent ostrine into estrine. So that's why the mice was affected with pneumonia. Even carbohydrate also, he did it, and carbohydrates he used. But even though when he used carbohydrates, the transforming principle was not able to change. That means carbohydrates are not genetic material, RNA was not genetic material, and these proteins are not genetic material. They confirmed it. Then heat-killed estrine, DNAs, they use DNAs, and live ostrine, and then injected to the mice live, was able to live happily. That means when he used DNAs, which was able to digest the DNA of the estrine, then the transformation of non-virulent ostrine into estrine was not occurred. And because of that, what happened? When it was injected to the mice was not at all affected with pneumonia, and it was very happy. And because of this, it has been confirmed that the actual cause for the transformation of the non-virulent ostrine into virulent form was DNA. And they concluded that DNA is the genetic material. So this experiment is very easy to remember. What they did, Oswald Avery, MacLeod, McCarthy, they worked on transforming principle, and they purified the DNA, RNA, protein, carbohydrate, etc. from the heat-killed S cells using suitable enzymes. Then what they did, they took some heat-killed S cells, and they used proteases which are able to digest proteins, and then after the proteases action, then they mixed that heat-killed estrine with R-strine and injected it to the mice. The mice dies. That means proteins are not the transforming principle. It was confirmed. Then they used RNAs once again by taking another colony of dead estrine, and they nullified the RNAs which are found inside the boiled estrine, and they mixed it with a live non-virulent estrine and injected to the mice. The mice dies. Then they used heat-killed estrine and DNase enzyme they used, which is able to digest DNA, and when they used it, after that they mixed that heat-killed estrine with the live estrine, and the mice was able to live happily without pneumonia. This confirms that DNA is the transforming principle. So all these experiments are important for 5 mark. They may ask. Okay. Now, let us see the next aspect, that is Hershey-Chase experiment. Hershey-Chase experiment. It is also called blender experiment. Blender experiment. Blender experiment. What they did, they made 2 preparation of bacteriophages. In one, protein were labeled with S35 by putting the medium containing radioactive sulfur. In the second, DNA was labeled with the radioactive sulfur 35. In the second, DNA was labeled with P32 by putting the medium containing radioactive phosphorus P32. So, they made 2 preparation. They made 2 preparations. What they prepared, let us see. Of bacteriophage, which kills bacteriophage. So what they did, in one, proteins were labeled with S35. In one, proteins were labeled with S35, radioactive material or radioactive sulfur, with the radioactive sulfur 35. In the second, DNA was labeled with P32. Medium containing radioactive phosphorus. P35. That means he made two preparations. In one, what he prepared, he made a medium containing radioactive sulfur S35. In the second, DNA was labeled with P32. P32 means radioactive phosphorus 32. Then after this, the preparations were used to separately to infect E. coli. After infection of E. coli, cells were gently agitated into the blender to separate the phage particle from the bacteria. So what they did, they used centrifugation. I will do it, I'll explain it in detail in the next video. But you remember this much, the Hershey and Chase, it is their experiment is also called a blending experiment. In this experiment, they made two preparations of bacteriophages. In one, proteins were labeled with S35. Proteins were labeled with S35 by putting the radioactive sulfur 35. And in second, DNA was labeled with the P32 with the radioactive phosphorus. So what they did, how they continued their experiment, I'll explain in the next video. Okay.